Good morning, everyone. I'm Chris MacDonald. I'm the Vice President of Data Science, Engineering, and Operations at Chromocode. We're a molecular diagnostics company. I'll talk a little bit about what we do as we go through it, but I'm here to talk to you today about our approach using data science for product development, field monitoring, and understanding how our products are performing. Normally, when you think of traditional QPCR, you think of a signal that looks something like this. You have a signal that amplifies across a couple of cycles, you have this amplification, and then it levels out at some point. Using this, you detect whether the signal passes some threshold, and if the threshold is passed, you would call that target detected. This allows you to put one target in each color channel of a QPCR machine. So in the case of this cartoon over here of this 4-channel instrument, you get up to 4 targets that are detected. For Chromocodes technology, what we do is we also encode the target using the amplitude of the intensity of the targets. So if you can imagine a fictional target 1 here, that when it's present in a QPCR reaction, amplifies to 50 fluorescence intensity units. And then a target 2 that when it's present in a reaction amplifies to 100 intensity units in the same channel, in channel 1. Then if it's at this 50, I could tell that it's target 1. If I read 100, it's target 2. And if I read 150, I know that targets 1 and 2 are present. By arranging the targets, the intensities, the concentration of the molecular components at different amounts so that we get a unique intensity for every target and every combination of targets, lets us resolve 3 or 4 targets per channel in a QPCR reaction. This enables on the order of 10 to 20 targets in qPCR and up to 50 targets in digital PCR. The way that this is all mapped out, we call the code space. And so when we have a target level 1, 2, and 4, these black ones, our targets 1, 2, and 3 mapped there, we call that the code space. As I mentioned, what this allows us to do is on a 4-channel instrument, as an example, getting up to 16 targets per channel. And then on a digital PCR instrument, we have a higher amount that we can encode, getting up to 50 targets per channel. And this allows us to create a lot of panels that we're talking about, but also this means that we have a huge burden in development to create these highly multiplex assays. And so what we're going to talk about throughout this after we get through the introduction is some of the ways that we leverage data science tools in order to enable this technology. Key components of our assay is that we are a digital or PCR and digital PCR assay development company. We sell the reagents. We also utilize an interlock data cloud for analyzing the data. So you upload your data to the cloud, you get back a report that tells you what targets are present in that sample. This keeps costs low for the labs. This is also instrument agnostic. So whichever PCR instrument you have on hand is the PCR instrument that you can use for this assay. Key features with this, then we have the reagent kits that enable the 4x capability on the qPCR installed base, but then using the software informatics cloud, you get data analysis and reporting for the customers, scalable network effect, as well as knowledge that feeds back to us to help us improve the tests, improve the customer experience, to be able to deliver more to those customers. What that ends up looking like here is that when you have a cloud, you upload the run to the qPCR run file to the cloud, the automatic analysis of the results on that cloud infrastructure, and then the visualization gets delivered back to the customer and the results are reported. At this point, we have a database of over 5 million tests with over 825 million data entries that we've done primarily from our COVID testing assay. Of the 5 million tests, probably the majority of them are from that. 2 big uses that we have on the data science side that we use for this really is number 1, helping develop real-time lab or customer data. So being able to collect and manage this highly multiplex diagnostic data to provide either epidemiological insights, understanding and helping customers understand their inventory, their workflow, their efficiency, and helping customers support being able to zoom in very quickly online and resolve any issues that are happening with customers has been incredibly valuable for us as we've built out this cloud infrastructure. In addition, anonymized aggregated data is something that allows us to take a look at these chemical signatures from the qPCR and do things like supply chain monitoring. We always want to ensure that we have the best possible production and performance for our different targets and our different assays in there. So monitoring the supply chain quality, using this to improve our assays, also for public health implications. As an example, we all know that strain variants happen from time to time in coronavirus. Being able to stay on top of that and ensuring that none of the strain variants are impacting our assay, or if they do, being able to detect that and improve our assay and also know that there is a local strain variant that's potentially impacting the assay can be of huge importance. We haven't seen one yet, but we're on the lookout out for it on a daily basis. As far as the different applications that we do and that we're really interested in applying all of these tools for is really understanding a lot of the applications around where you use NGS. NGS currently is really great for biomarker discovery. We'll talk a little bit about oncology coming up a little bit later. Their instruments tend to be expensive, the workflows can be complex and slow, and there's a lot of data that you have to work through to compute your answers. On the contrast, with qPCR, it's simple. There's cheaper instruments, it's faster, and it's very easy to compute the data. If you think about qPCR, it's often a single portion of the workflow of NGS. You might have an amplicon sequencing product where you're looking at a specific set of markers. So NGS is great for discovery, but when you know what you're looking for and you can reduce the amount of things that you're looking for, PCR can be a valuable tool in someone's arsenal to be able to understand what targets are present in the sample or what to do about something that's happening. The idea here is that if you look on two axes, you have one axis, which is the speed to result or the cost efficiency of being able to get to an answer about some particular sample, and then also the amount of content that you get. There's a cost and content gap where the amount of high content results, where the speed to result is very important, provides an avenue for us to go in and inhabit and try and provide the answers that can only be done with NGS. In general, data science approaches to being able to do this and compress information into different results is not new in the life sciences. There's been quite a few different companies that are involved in various different areas in life sciences. So from PCR AI and Genentech, automating PCR analysis, improving accuracy, using artificial intelligence and digital pathology, whether it's IBEX, PathAI, Phage, or Protea, as well as machine learning for single cell analysis. BD Epic is doing a lot of work down here in San Diego as well on that side, as well as Clever Culture Systems, and innumerable companies are exploring this. One of the interesting things I learned when I got into the biotech area is that although it's a very different subject matter, the models really are the same when you look at the applications of data science. So if you might be in the tech industry and you're doing image analysis algorithms to try and classify images, you might have a problem to look at something like this. You have this picture here, you're trying to determine whether this picture here contains a cat or a dog. So what you do is you take this image, you train it on some either deep neural network or other model, which has an input layer, some neurons, and an output layer, and then you go through that decision process. And then the final output of this is that you see a dog present. The data science workflow for image classification, for serving advertisements, for analyzing cells to determine whether there's pathological cells in the sample, to analyze NGS data to determine what base pairs are present or what mutations are present, really fundamentally are the same. So while the neural network might be trained on something different, the whole process around going through the data science steps to understand what is done is the same. The core data science workflow that anybody's going to undergo typically has four major phases, are all divided up. The initial phase, really where you want to decide on the problem you're trying to answer and acquire some data about that problem, whether it's an image of a cat, whether it's a slide or a histology slide that has some cells that you're trying to classify. The same idea you start with and then you put down into your pipeline. The next step is called pre-processing. This is where you clean the data. So you may have data that is useless. Maybe some of the slides are broken, maybe some of the images aren't really usable. Then the next step you do is you extract features. So in the case of a facial recognition, the features you might extract are things like the distance between the eyes, the size of the nose, the size of the mouth, the different features around the face. And then you can use that to compare one image to another. In the case of a cell, you're looking at the number of cells, the size of the cells, the shape, color, if they're stained, and things like that. Or in the case of qPCR, we're looking at features of the different curves. From that point, once you've extracted the features, you go through a process called feature engineering. This is taking a look at those features and trying to figure out what's important about the classification that you are trying to do. So again, in the case of facial recognition, maybe the distance between eyes is a very important feature that lets me recognize phase 1 to phase 2 to phase 3, but whether the shade of the skin isn't as much a differentiator because many people look similar in that respect. After that, you're going to have some missing data. Some pictures are incomplete, some faces are done at different angles, some cells are missing from different ones. Once you figure all that out, you're done with the pre-processing step. At that point, you generate a model. So the models you do then try and make some prediction about what I see. So under these certain parameters, under these certain circumstances, I think that this is going to be a cat, or this is going to be a dog, or this cell comes from a tumor sample or a normal sample, or it's pathological or it's not pathological. You try this in many ways. You might do a neural network model. You might do some more simple linear models to make a decision on these parameters. And then you evaluate and you select which model is giving you those best predictions. After that, during that modeling process, you evaluate the performance. You go get more pictures of cats or dogs or cells or whatever you need to take a look at. And then you see, does it predict accurately on this new data what I'm trying to do? And if the answer is yes, then you can then take that model and put it into production to go get answers. Again, in the case of image classification, your Google search or your Facebook image tagging. And in the case of cells, trying to predict for a new sample whether that contains some pathological cells, whether they're tumor cells or something of the like. And in our case, whether the PCR signals have target 1, 2, or 3 that are present in a particular sample, and how you deal with other features of the data set, like the wells of the PCR plate, or the non-uniformity of lenses, or other things that we need to correct for in order to make sure that we get the right data every time. Sometimes you're going to go through this process, you're going to evaluate the performance, and it's not going to be so good. So maybe you get down here and you predicted the picture of it's a cat, and it's actually not a cat. And so what happens then? Well, in the case of big data or machine learning or image classification, the typical way that you will go through it is to go and get more data. And so this is a pretty typical approach. This is what you'll see in Google or something like that. If you have hundreds of thousands of images to classify from, there's a huge and rich data source to get that data from. So you take that data and then you go back, and then you're able to provide more data, and then go back through your evaluation of the performance, and then get through all those different pieces. So this is critical, and this is really important when you're trying to evaluate your model. You go back through it and you get better answers. This is also an example of how you might work for analysis of the self-driving car data. These Google cars, Tesla cars are out here collecting more and more data as time goes, refining their algorithms and getting better and better and better. The key thing here, though, to understand is what this means, that in big data, the company that has the most data is the one with the edge. An example here is Grail, which has done a fantastic job looking at many, many samples and trying to understand, how do I detect cancer early using just the DNA that's in the blood and looking at markers that are in there. This was a huge fundraising process to get in there and get tons of samples and look at a lot of data with just incredible results that they've had and really great outcomes for the company. This is great. And for companies that are able to do that, this is really powerful. That's not necessarily true of all companies, one of them being ours, where we can't necessarily go and get more data than anybody else that is out there. So what do you do? So if you go and you come up with the wrong one, but instead of being able to get more data, another approach that you could potentially have that might differentiate this is maybe you can go out and generate your own data. So when I spoke of feature engineering before, in the classic sense of the data science loop, what that really means is determining what features are available that already exist in that data. For example, the eye width is predictive of the facial recognition algorithm, or for cats versus dogs, maybe it's the ear shape, as an example. Well, in the case of for us, when we design QPCR assays, and in the case for a lot of biotech companies, you're actually creating the chemistry that creates those features. We design TaqMan primers and probes. We design the chemistry, we design the dyes, we design the image algorithms. And so in the sense that what we can do is we can use feature engineering, which actually means a biochemist tweaking some assay or trying to decide, does this need to be brighter, does this need to be dimmer, do I need to switch the dyes out? And so we call that literally also feature engineering. And in the case of facial recognition algorithms, if I could re-engineer a cat so that they always had a green hat on top of them, then it would be very easy for me to distinguish a cat and a dog. All I have to do is look for the giant green hat, that I don't have to worry about whether the facial distance, eye shape, or the nose, or anything like that is predictive always of every breed of cat or dog, because I have a very obvious and a clear feature in which to train my data on. So qPCR provides the right field for being able to do this. If you've designed an assay before, you have control over the dye, but not only the dye, but all of the components within the reaction. You know, you have your DNA sample that comes in, you have primers, probes, nucleotides, your Taq polymerase or other components on the enzymes. You have your buffer, your small molecules, your salts, the type of tube you make, the thermal cycler, the cycling conditions. You have this huge parameter space with which to operate. And typically with assay development, in order to get the PCR signal. Now, in the case of really the data scientists, getting them in the loop early so that you can take a look at those data and try to understand, well, hey, you know, if you could do this signal a little bit brighter or dimmer, if you could do this or you could do that, that increases the resolvability on the end of what you're trying to do, then you're going to really be able to drive to a solution that is much more distinguished between the different results that you might try and do. And so in the case here, right, so if you think about the PCR, we started with a simple PCR curve, reaction initiates, amplification, reaction terminates here. You get one piece of data out in the case of, let's say, a SARS-CoV-2 assay that we have currently on the market, or any assay that you might be developing, right? So what you're getting is you measure 60 floating point measurements. I get 60 cycles worth of data, or 40 cycles, or 20 cycles, or however many you're doing. And you get a number that's down to, 5 decimal points of the different amount of fluorescence intensity that you get. And you reduce all of that data down to one thing, whether the SARS-CoV-2 is present. In the data world, or in information theory world, you call that one bit of information theory entropy, where either my target is present or my target is not present. What that also means is that you're throwing away most of that data. So as you go and you start engineering the qPCR curve to be able to generate different features for different amplifications, you might have one target that amplifies very early at a very high concentration, and then a second target in the same channel amplify later in a second amplification. What that looks like is two PCR curves stacked on top of each other on the left, and maybe those are operate totally independent from each other, and the data works great, and then your assay is going to work out of the box. But sometimes they're correlated with each other. So using the data science tools in order to be able to determine why are they correlating under what situation, and feeding that back onto the biochemistry team allows you to dictate things, using the enzymes, the salts, really all these different components dictate the curve shape such that then your features can then become independent of each other. Circling back to the initial slide that we started with, that's really how you build up to assays. In our case, what we're interested in is compressing information into qPCR signals. So you can get 16 targets in a 4-channel qPCR system, 50 targets in a 5-channel digital PCR system, as an example. So we're interested in utilizing those tools in order to compress more information into the same qPCR signal. In other cases, ethic sciences or other image recognition algorithms, you're talking about looking at different stains or different images to try and extract features. You also can do this exact same loop, where you're trying to extract these features from it, but you also have the ability to perturb that system, whether it's through choosing, dyes and Taq primers and probes, in our case of qPCR, or in the case of some cell slide, looking at how you stain them, looking at what kind of, what you attach to it, what markers and things that you do there. What does this result in? Well, here, as an example, we do have several infectious disease assays on the market, but another way that you can look at it, right, is here we have a proof of principle. This is 3 markers in NSC, non-small cell lung carcinoma assay, that is under development to be able to look at multiple markers within a single well. So in this case, this is looking at 3 markers in a single channel on a qPCR instrument. Building that out allows us to generate the sensitivity with qPCR. In qPCR, you have down to 1% minor allele fraction, but then in digital PCR, you can get even significantly lower than that as well for things, applications like liquid biopsy. Then developing that and expanding that allows you to generate full coverage across the 15 to 20 markers that you might need for a full non-small cell lung carcinoma panel, just using qPCR or digital PCR. The value here for us, an interesting side is that using the techniques of information compression, you're able to generate a full coverage and with the time to result of hours without needing a central lab for a very low cost of sample. And this all goes back to that observation that when you're looking at a sample and you don't know what you're looking for, of course, you're going to need to sequence. You're going to need to look at a huge depth of information to try and decide, what do I do about the sample and what do I do going on in the future? When you know what you're looking for, and the subset of markers is really only 10 to 20 to be able to guide the decision or be able to understand what you're looking for in a sample, then you can start to do that in the qPCR or digital PCR space and start to generate a lot of value. Because what we're here for is really to improve that for our customers, but also ultimately for the patients that want to use this technology. So taking that, running that all the way to the ground, all the way at the end, you're trying to look at several different things. And this is where the development gets a lot more complicated, and those tools that we talk about get even more important. You're trying to get signals that get generated, whether it's a signal nucleotide variant, deletions, insertions, fusions coming on, and these different things that you're looking for. Sometimes there's 8 different RET fusions that might happen, 8 different ROS1. Some of the, EGFR exon 20 mutation insertions, there's multiple that you see as well. Exon skipping deletions as well. So there's a lot of information that come out of this. And so utilizing those tools is critical. Otherwise, we wouldn't be able to do this development. So the key here is really integrating the data science team into that development, as opposed to just having it at the end of the cycle where you're using it to try and understand product performance or performance in the field. And we'll get to that as well in just a moment. But to summarize this part of the talk, I think the key here is to really put that together. Taking control of that data structuring allows new applications on existing tools. In our case, our interest is digital PCR and qPCR, and we think of chemistry as the opportunity to structure that data. So it's a data structuring activity. The qPCR instrument converts that data into some digital signal that has an output, and those outputs we engineer to be distinct depending on what targets are present. And then we're able to do the science and the analysis in the cloud to do things, enabling respiratory virus marker panels, antibiotic marker research, information on chromosomal aneuploidies for not prenatal samples, and delivery of oncology mutations or information on those that are present in the sample very, very quickly. This all doesn't happen in a vacuum. We talked about the cloud over here, and so it's also key and important to build up the infrastructure that allows us to do the analysis both on our internal samples, but also be able to support this in production as we go to customers. And so while there's a range of qPCR instruments, they'll go and then they'll upload the data, we get the analysis, and we export the test results back. It's really key that we develop this in a manner that is not only effective for us, but also secure and private for our customers. So you need to develop this out. In our case, the Chromocode Cloud is a fully HIPAA-compliant solution that does the instrument characterization and analysis, visualization, reporting, and has secure and scalable data management. We do this through architecting the cloud and dividing up the part where we do the data management, the actual analysis, the visualization, and reporting in AWS in a HIPAA-compliant environment, and then the data upload and then the test reports for the customers come in and out of it, but we're able to lock that down and keep that secure. This allows us to do several things. Number one is allow us to see what's going on in the field. So for us, we're not so interested in what a customer's doing. That's their job. We want them to be able to access data to help themselves. But we do want to know things like, as an example, here's the COVID positivity rate testing. And so we can see if the positivity rate of COVID is going up or down, or what's going on with it on a daily basis. And so that helps us understand, is there a variant that's coming that's going to need us to spin up operations? As an example, are we going to need to be making more tests to support the field? How is it performing? Do we have one of our targets dropping out, an S gene target or an N gene target to, that may be indicative of a new variant that we have to respond to quickly? Having that data in real time is really powerful. This also helps us understand the inventory. If folks are starting to use more tests, we want to be ready to also give them more tests so that no one runs out. Availability and supply chain has been a huge issue for the last 2 years throughout the COVID pandemic. And any edge you can have on predicting what's going on allows us to keep stable supply for our customers and allowed us to never go on back order on our tests once throughout the last 18 months or so that we've had this on the market. And that's been both huge value for us, but also really the important thing is supporting customers, and ultimately they're supporting patients and running these tests that enable them to keep a consistent standard of care in the communities that they serve. In addition to that, we have work on going forward, trying to understand what kind of epidemiological insights. If someone's viewing this talk and is interested, please reach out to me afterwards, because we're trying to pursue, there's data that says, looks at, for example, sudden increases in viral load in certain populations can be indicative of a new strain variant coming out. And that may give you a heads up or an advanced notice that something's happening before all of a sudden 99% of the population is infected with a variant like the Delta variant, as an example. And so trying to extract data on this aggregated data of millions of samples is something that we're looking to do, and definitely interested in talking if someone is interested in pursuing that as well. And then finally, software support and monitoring. We want to ensure that our customer experience is as good as it possibly can be. And so we want to ensure that the uptime is 100%, that people can use the test whenever they want, and that we're able to serve them with tests as soon as they possibly need. The benefit here is that we can do this all over the cloud, and so it's easy to ship someone a kit, Zoom them to do the training, they can upload the data and be running with their tests the next day. Finally, as we start to think about the assay, now that we've designed the assay, we've engineered the assay, we've developed cloud tools that can help us understand what's going on with the assay, we want to be able to use that and leverage that to improve our products. As you look at maybe a reason that an assay wouldn't perform as well as you would hope, there's so many different sources of noise, whether it's common cause noise. Typically this is generally normally distributed, commonly characterizable phenomena. This is common cause variation, right? Maybe plate to plate, reagent to reagent, lot to lot variation, instrument variation, clinical sample variation in the case of our EUA assay as an example, or what we call non-Gaussian variability. This is something that might be non-normally distributed variation. So this is a good example of that I already mentioned, is a strain variant. We might be concerned there might be a strain variant popping up, or maybe sequence interaction, or something that's under the LOD, or interfering substances, or something like that. So because of this, there's so many different sources of variability, it's impossible, although the process for getting one of these on the market is quite rigorous, it's impossible to have everything fully understood. New variants can come out over time. That's really where the power starts to really exercise itself. Here's an example of over 30,000 samples of a COVID test over a 6-week period fairly early on in the pandemic. The ability for us to monitor, first of all, put 30,000 PCR curves on top of each other in one plot, helps us understand how well the assay is performing, as well as the lot-to-lot variability. This was done early on as we were starting to produce and starting to ramp up. This was over 3 lots of raw material that we got from our suppliers and we're shipping out, and enabling an understanding of that endpoint intensity, that the variance between lots was very, very tight and showed no significant differences, was really powerful as we get these new products to market. And we're all ramping up at the same time, trying to serve this, in this case, the COVID crisis, being able to understand that the performance was good over time, and helping to make sure that that is true, and across manufacturing and supply chain issues and things like that, has been really valuable for us over the long haul. So in summary, there's many data science tools and methods that they enable increased accuracy and robustness from the upfront product design, and that can open new areas in development. This is what enables our new technology and new market areas that we can address with qPCR that you can't, and enables folks in the cell image analysis to open up new market areas as well. So whatever you might be doing with your company, there's a lot of valuable tools there that can help you. Real-time monitoring, operational insight allows us to keep our customers fed with tests so that no one runs out and anybody who needs a test can get it. Allows us to also improve the product and marketing or monitoring. And then finally, really investing in developing these data science tool sets up front in your organization leads to these huge benefits down through product development and operational execution, all the way down as you go. And then once you're on market, that starts to keep snowballing to get higher and higher benefits as well, as you start to learn more about your products, as you start to learn more about how to stabilize them, how to increase the robustness, and then ultimately launch better products with more density of targets, in our case, down the road as you go. All right, well, thank you so much. Please feel free to reach out to me if you have any questions after you've viewed the talk, and thanks for joining me today.